Vou colocar o arcão dele.
Carlos, você pode aguardar eles lá na recepção? Faz favor.
Professor, fica à vontade, tá? É só ligar o microfone aí.
Bom dia, testando.
Perfeito. Já estamos online, já, tá? Transmitindo ao vivo.
Daí para apresentar eu.
Abre o PowerPoint e deixa aí a apresentação. Se o senhor quiser até sentar ali no computador, não tem problema, tá?
É que eu vou ficar e daí vou chamando os professores que vão apresentar.
Tá bom. No momento nós temos 2 pessoas assistindo. Então, eu acho que eles estão ainda também se chegando.
Deixa eu esperar o dele.
Não, na verdade, o senhor conseguia abrir. Conseguia abrir.
O sistema no computador do professor Maurício, você já acessa também.
Mas na verdade ele já está aqui, né, online. Eu pego o computador dele como se o computador dele estivesse apresentando. Então, na hora da apresentação, vocês vão estar com essa câmera aqui, né, olhando para a câmera, apresentando aqui, mas vocês vão estar passando o slide por lá. Eu consigo colocar o rosto de vocês e a apresentação, eu consigo colocar só a apresentação. É verdade.
Aí vai jogar a tela também tela do computador nas TVs também.
Good morning. It's an exciting moment for us today, receiving our colleagues, scientists from Japan, that it's unnecessary to say the friendship between Paraná State and Japan. In the past, several immigrants from Japan came to Brazil, and looking for opportunities. The ascendants of Dr. Tomoro Seira, for example, and they give a huge contribution to Brazilian agriculture. The Japanese and their descendants. Dr. Tomoro is a great example in crop systems in Brazil. And today we will be talking about cooperation and partnership, that the science specifically is a sector of the society in which these words are extremely important. Beyond the great laboratories, intelligence, cooperation, partnerships is fundamental to great breakthroughs in the science. And we would like to ask the scientists that we will be discussing today can present themselves, the field that they work in, the university. Okay, Dr. Watanabe, maybe you can command your group, please. No problem. Very good.
Good morning. So let me speak in English, so the easier for me. We are a group with the several different Japanese institutions. So University of Tsukuba organize this the delegation. So we have Professor Tamura from the Kagawa University here. He is the food scientist, and Mr. Nagata from the JATA, which is the under Ministry of Agriculture for coordination, especially for international aspect, under the Dr. Kanamori, Jilkas. And Jilkas known to be very active. Yes, he lived here 6 years, and he used to work on soybean breeding. So we have some initiation already with the state of Paraná. And one more, myself, the first time I came to Paraná was the 44 years ago when I was a student. So I came by Curitiba, passed by Londrina, Cascavel, then I went to Foz. Principally purpose was to get to know about your agriculture, that it would be very the benefit for young man, me, to understand the big and the dynamic agriculture. And now it is really true that you have a lot of success. So we'd like to learn how you would proceed and how we could see a common interest of the research and application activities. Thank you.
We now may initiate the presentations of we try to give you a short outlook of some projects in university that we are thinking they are candidates to cooperation with Japan. And first of all, of the presentation, Dr. Caldarelli will be presenting some aspect of the apiculture chain in the Paraná state. And the next will be 3 presentations regarding using inputs in agriculture, mostly bio-inputs and nano-inputs. And there is an interesting history about this question. Brazil present is the world leader of bio-inputs in agriculture. And we have to say thank you for Japanese people that brought in the past the bokashi compound to be used in agriculture. And we worked a lot with this material we use for low-income farmers in Brazil. In our research group was the group the most worked with bokashi compound in Brazil. This is a very interesting story about the contribution of the Japanese that came from Brazil. And the third subject will be functional foods that Dr. Carla will be presenting for finish the morning. I would like to say that if you have any question during presentation, you could interrupt and to make more informal the presentations. Then we start with Dr. Carlos Caldarelli.
I think in the schedule at the end of the Brazilian presentation, we have a short speech by one of them. And I'm sorry, I forgot the important person. So Marlon Rivera is my student. He is assistant professor at the University of the Philippines, Los Baños. So you are not only Japanese, actually international delegations. Okay, thank you very much.
Good morning. Just one moment. They are preparing the presentation.
So just to use this time, I think
For a brief.
Paula, she's from Quer, and she's worked with engineering, food engineering, right? And she's also with this joined this mission since we depart from January on Foz
Oh, welcome.
My name is Jackson. I'm from Paraná-Japan. I'm working with the inputs. So we are conveying the mission since Saturday.
Good morning. Okay, welcome. We are very happy with this mission here in Tuel, and it's a very auspicious moment for our project because they are Alcara Foundation. Now it's start with the new arrangements, new research and innovation arrangements, NAPIS, and they are open several opportunities in different areas in agriculture, in different areas of science, and it's a very special moment for us. For example, my presentation is about NAPI bees, about bees and beekeeping chain. And for us, it's a very special moment because we are preparing the activity for the next 3 years. And we are open for partnerships and for new opportunities. We are very happy and welcome to Well. Ok? É uma que tem uma abelha, uma flor grande. Ah, esse. É esse. Just one moment. Okay, officially start. Good morning, and thank you for the invitation. Really thank you for Professor Mauricio. It's a pleasure for me to share our research with this group and introduce our team. Firstly, I would like to introduce myself. My name is Carlos Caldarelli, and I am a professor in the Department of Economics in this university. My major is economics, and my area of expertise are rural economics, economic development, and macroeconomics. And for a long time, I have been working with some colleagues in agronomy and agrarian science. And this project that I will present today is the result of this collaborative work for a long time. Not a long time, but a long time. Today I will present, please, pode passar, por favor. Okay. I promise that I will be very strict with the time. Professor Mauricio told me 15 minutes, and I will be strict with this. Today I will present NAPI BIS. NAPI is the New Research and Innovation Arrangements, NAPI, from Araucaria Foundation. The Araucaria Foundation is an important funding agency for research in Paraná. And this arrangement, this initiative, is formed around different networks of science. And in this case, we are with 7 universities in Paraná, and the subject and the main subject of our research is bees and byproducts. For example, royal jelly, honey, pollen, propolis, and wax, and very especially the most important, pollination. And for our region, geographical region, it's a very, very important issue because for the agriculture, this is a special question with a huge literature about pollination and bees for the production. The topic is crucial in Brazil, especially in southern region. Rio Grande do Sul is the large producer of honey and different byproducts. I was talking with professor here, and he told me about the Japanese culture with propolis and bees, and for us it's very interesting because we really don't know about this one. The scientific research are really interested in pesticides, bee mortality, and the effects of climate change. Pode passar, por favor. Pode passar? Tá bom. As I told, we are in different institutions, 7 universities in Paraná, more different partners in the production, in different institutions and founding agencies, and also with some representative market institutions. The NAPI BIS, it's really huge NAPI, and the project is organized into 5 axes or 5 great key areas that I will present. Please. The 5 areas, this is our overall of our project framework. The arrangement work is divided into thematic axes, 5 key thematic axes. The first one is the risk of pesticides to bees, and this is a really important question for our region, and a huge literature about this question is built in Brazil. The second one is the characterization and quality of beekeeping and the meloponical products. It's a very specific kind of bees. The third is the genetic improvement of species, and this is really important for the quality of bee and the different resistance of these animals, for example, to pesticides and to honey production. The 4 business schools, it's about marketing and the quality, the relationship about quality and the marketing. And the 5 thematic axis is diversity, ecology, and conservation of bees in Paraná. Here in our group, our research group, based at Londrina State University, is involved in business and economic development, the X number 4. And I will present very briefly this thematic area and our research is in this area. Please. Business school and beekeeping value chain objective is integrated the knowledge of the production and the marketing. And the main objective is the support to beekeeping and meloponical production and added value and marketing. In our specific axis, we have 3 three key issues that I will briefly present, please. This is a figure to represent the complexity of the key element and the key elements in the beekeeping chain. And we are integrated with pharmaceutical, food technology, genetic, agriculture, and this is only to illustrate the complexity of our subject. And business and economic development axis 4 would like to study specific the relationship of this one with the marketing. Please. In this way, our focus are first in quality, and in this area we aim to better understanding the beekeeping value chain and how improve product quality. We also investigate origin certification, quality certification, and quality standards in this area. A key element is the legislation and adequacy of the production to Brazilian legislation. Two important upcoming events related to quality are scheduled for this year and the next year. The first one is the Bee Day in May here at Londrina State University. The Bee Day is a big event with scientific research and different partners and also the producers from all of Paraná State. And will be in May here at Londrina State University. And the second event is the Honey Quality Contest in the first semester of 2026 in partnership with the Brazilian Ministry of Agriculture. And the objective of this event is to establish a standard for honey quality. For example, as we have in coffee and cocoa market. And this is the first main axis of our research group. The second one is about added value, and this axis explores how quality certification and regulatory compliance can increase the added value of beekeeping products, ultimately improving profitability and the small producers. In this aspect, the Bee Day, for instance, brings together various partnerships and facilitated connections between producers and marketing. The third one is certification, and for certification, we need to establish standards for honey and byproducts. And for us, it's very important because honey, it's a question very important for the production of food and food technology. And byproducts, for example, as propolis, royal jelly, need to establish quality patterns and standards for commercialization and a better integration with marketing. And the third point is sustainable production. And in sustainable production, here we analyze key factors such as climate change and the impact of pesticides on beekeeping industry. And we focus on the interconnection between quality, productive, and economic value. And our group is studying how we can organize and structure here at a laboratory for honey quality, and how to establish standards for quality and certification that it's not better established in the Brazil. The beekeeping value chain is highly complex, as illustrated in the figure, and the collaboration of economics, agronomy, biology, and food technology can open new avenues for advancing in these fields in Brazil. This presentation, please, This presentation comes at an exciting moment as we are developing our plans for the next 3 years of our project. And at this moment, we are open to partnerships, alternative approaches, and comparative cross-country analysis. And we are very excited and to better understand how this chain is organized in Japan or in other countries. I am very pleased to introduce our team. Myself, Professor Carlos Caldarelli from Economics Department, and I am the head of this project at WELL. Professor Vilma Espinosa from Food Technology Department and Agrarian Science, and Professor Silvia Sofia from Department of Biology. Along with our undergraduate students, graduate students, and 3 postdoctoral research. It's very important for us to share our research and our group. It's a very huge project, and we are open in different axes for partnerships, and very especially in the Access Business School at WELL. I would like to highlight that the other axes are each axis is organized in one university: Maringá, Cascavel, Dois Vizinhos, Federal University in Dois Vizinhos, Federal University in Curitiba, and here the access is Business School, Quality Certification, and byproducts from bee and honey and quality. Thank you. I think that I am in time. 30 minutes. We are open for questions, and we are very happy to present. Thank you, and this is my contact for the future. Thank you very much. Professor Maurício.
Okay.
Please. Thank you very much for your presentation, and I would like to congratulate you to make this study multidisciplinary scientific team. So we don't see particularly on bees. Although we have a lot of pollinator problem in agriculture, like a tomato production, cherry tomato, we need bees. And also strawberry production, we need bees. And generally fruit trees, cherries, and those that we temperate countries, we need bees. But we don't have bees. The bees are gone. So then they have to buy bees somewhere. And American bees come. Then Italy invade also our ecosystems. So honeybee, Western honeybee, American bumblebees, honeybee, so they change. And the Japanese is gone. So we have a double problem of ecosystems. But I don't see much of the effort how to recover local bee species. Just we are carrying some errors, but not much stability. So it is true that not only inside the home, but in population, in agriculture, they are speaking of pollinators, insect pollinators, bees, flowers, also pollination, conservation of biodiversity. I think that Japan faces a lot of progress, but not likely we are not yet happy with that transition. Probably we can learn.
Thank you. Thank you, Dr. Caldarelli. Next, we will have the presentations of input sections, and I invited Dr. Halley to show their projects.
Tudo bem, tudo bom? Eu vou falar os dois. Vou falar no lugar do André também. Não, vou dar duas em um, vou falar. Ah.
Daí tem essa sala que está conhecida, aí aparece informação do pessoal para ver como
Vou dar mais rápido.
Pode registrar a presença aqui dos estudantes, acho que é certo, para enviar um certificado.
Ah, não pega ali. Acho que não passa, né? Não tá passando. Ah, não passou. Okay, so good morning. I am Professor Ale Caixeta de Oliveira from the Department of Animal and Plant Biology here at the State University of Londrina. And it's a pleasure to be here and present some of the studies that we are doing in the NAPI biodiversity. I am one of the articulators of NAPI biodiversity in Paraná State. And it's as I think the previous presenter already showed you what a NAPI means, the new arrangements in research and innovation. So in the NAPI biodiversity, we are more than 100 researchers from the different universities of Paraná State. And we evaluate different aspects of biodiversity. Today, I will focus mainly on the use of biomaterials and associative microorganisms to improve plant growth and stress resistance. I will speak also on behalf of Dr. André Oliveira. We will have now two presentations in one. So I collaborate with Dr. André, and I will talk on behalf of him also. Okay. In my presentation. Oh, okay. Okay, so I will talk mainly on our research lines regarding plant and microbial response to abiotic stresses, and also about the nature-based solutions to improve plant growth and stress resistance, especially the use of associative microorganisms, the nanotechnology, and also composite biomaterials. So the NAPI biodiversity is this huge research network. It's divided in different NAPIs. We have the Restore that's focused mainly on reforestation. So it's nature-based solutions to improve reforestation, and also the NAPI genetic resource and biotechnology. It also focused not only reforestation but also in agricultural and silvicultural applications. And also we have here the projects, the National Institute of Science and Technology in Nanotechnology, for sustainable agriculture, and also the long-term ecological research, the Atlantic forest of northern Paraná. Okay, so I will just give you an overview of the research that we do in these different networks. Next, please. Assim fica, is it better now? Okay. So regarding the plant response to abiotic stresses, so we test the response of 3 species, of native 3 species, and also crops to different abiotic stresses, like light stress, for example, highlight, or also shade stress, heat, high temperature stress, pollutants, and also drought stress. And so we evaluate both, as I mentioned, both 3 seedlings that are used for restoration, forest restoration purpose, and also different crops like soybean, maize, and other crops that are relevant to Brazilian agriculture. Next, please. Just some examples of this research. So in the Restore project, we are carrying out experiments with 3 seedlings native of species native to Brazilian Atlantic forest. And in this project, we have a collaboration. We have a joint project with Germany and also with France. So they are. It's not working. Is it working? Yeah. And so in Germany, they are working with the German temperate forest, and in France with the Mediterranean oak forest. So we are doing like comparative studies, ecophysiological studies regarding the response of 3 seedlings from these different forest ecosystems to drought stress. Okay, so I think this is an opportunity. So perhaps with Japan, if we could do like to join Japanese trees in this research, so we could compare how these different species from different forest ecosystems would respond to drought stress. And also in this study, next please, we are evaluating the responses of the microbial communities which are associated to these 3 seedlings. So we are doing experiments in the field, evaluating how the microbiome of the trees associated to the 3 seedlings, or to the trees in the forest ecosystems, or how they respond to the drought stress. So we collect samples in the wet season and the dry season, and are comparing how the microbiome changes according to the season in the different countries. So we are trying to find unifying patterns of plant-associated microbiomes in response to drought, to different humidity scenarios. And we are also carrying out drought simulations using the forest soil in the greenhouse experiments, and we are evaluating both the chemical and the biological traits in this soil, also the metabolic activity of the soil, and also some using out sequencing, some next generation sequencing to evaluate how the different microbial communities that are found in these soils. Next, please. And then we are also testing different nature-based solutions to improve the plant stress resistance. We are using associative microorganisms, polymeric nanoparticles, and also composite biomaterials to obtain 3 seedlings with higher quality for reforestation programs. And also, I think it's cutting the bottom of the presentation, but also crops that are more resistant to climate change by using sustainable practices. I will tell you some words about each of these nature-based solutions. So first, with the associative microorganisms. Next, please. So in this project, we isolated and characterized new strains of microorganisms, mainly bacteria, from natural environments. We did this here in Londrina region, in Atlantic Forest, and also in Ponta Grossa region. It was a different soil type, and we could obtain a library with more than 500 bacterial strains, and they were characterized both at biochemical and also molecular levels. So we have obtained many strains which are very promising to improve plant growth and also to improve plant resistance to stresses. For example, next please. We obtained some bacteria which increase the germination, the initial development of some species, of some tree species, like Cecropia paxitacea, which is a native tree species from Brazil, a pioneer tree that the germination is not very high, but using inoculated bacteria, it could improve the germination in the initial development of the seeds of these species. Next, please. And also we tested the effect of this bacteria in improve the development of seedlings in drought conditions. So we obtained, this is an experiment with tomato. You can see there that increase of the plant growth under drought conditions by use the seedling, the strains, some bacterial strains. So they are very promising in improve the plant resistance to drought. Next, please. And also tree seedlings. We observed, for example, this study was with Cecropia paxitacea. Also, we could observe that the inoculation with these strains, we increase the root dry rate, also the root to shoot ratio, and also the photosynthesis, the leaf gas exchange of the seedlings of Cecropia paxitacea seedlings under drought conditions. So we observe a higher resistance of Cecropia paxitacea seedlings to drought also. And next, please. And also we are using some fungal like Trichoderma strains. So this is an example of study also with Cecropia using Trichoderma asperellum, which was isolated from Araucaria angustifolia roots, native to species native to Paraná, to Brazil. And we could also observe that Trichoderma, the inoculation with this Trichoderma increased the growth and also the drought resistance of neotropical tree seedlings. Next, and finally, we are also not only evaluate the potential application of this technology, of these microorganisms, but also we try to understand the interactions of these strains with the plants. So to understand what are the mechanisms involved in the growth promotion and the drought resistance, like evaluate doing some anatomical analysis, metabolic analysis, and so on. So we are trying to understand the mechanisms involved also. Okay. Regarding the next steps, please. The next. The próximo. We are also using nanobiotechnology as a nature-based solution to improve plant growth and stress resistance. So we are using different type of nanomaterials, for example, green metal-based nanoparticles like copper nanoparticles, which are synthesized using green tea, for example, leaf extracts to produce these nanoparticles. So they are very effective in improve plant growth and resistance, at the same time that they are not so very low toxicity to the environment. We are also using some carbon dots, so we carbon quantum dots. In this case, we use different agricultural residues, wastes to synthesize these carbon dots, like residues from swine production, from sugar cane, and other residues. So to produce these carbon dots that can increase the photosynthesis of plants and also their stress resistance. But I will focus here mainly in the polymeric nanoparticles. We have many studies that we are focused mainly in polymeric nanoparticles. So in this case, we use the nanoparticle to encapsulate agrochemical, to encapsulate an active ingredient. For example, you can encapsulate herbicide, insecticide, a fungicide, and also a plant growth regulators. And which is the idea of this nano encapsulation? So it would allow a controlled release of the compound Also, it could protect the compound from degradation and increase the solubility in some cases, and so we could have an improved efficacy, and also at the same time it has a decreased toxicity to non-target organisms. The next, please. So, for example, we have some studies with herbicides. The example with atrazine, loaded polyepsyl caprolactone and zein nanoparticles. In this case, by encapsulating the herbicide, we could decrease the dose of the herbicide by 10-fold. So we could reduce the dosage from 2,000 grams per hectare to 200 grams per hectare, maintaining the efficacy. So we could control the weeds while decreasing the environmental contamination. We also have, regarding the insecticides, we have many studies using botanical insecticides. For example, the neem oil, that in this case, so they are they show very high efficacy very high efficacy in controlling insects and also as insect repellents, but usually they have a problem in solubility. And by encapsulating this neem oil, you could improve the solubility, and also it can protect the neem oil from degradation, for volatilization. So it could improve the efficacy of this botanical insecticide by using the zein nanoparticle. And it's interesting that zein is a protein made, so we can have a nanoparticle that are made only from natural compounds. And also we have some experiments with some fungicides, for example, with copper chitosan nanoparticles. So we can use chitosan nanoparticles to encapsulate copper and to control some fungi. So we have some studies with coffee, to control some diseases in coffee, in collaboration with Gustavo Cera from the Agronomic Institute of Paraná. But I will focus here in my presentation mainly in the plant growth regulators. The next, please. Because mainly biostimulants, many plant growth regulators, when we treat with the plants, they can degrade very quickly. So you need to keep treating many times. You need to treat many times with these formulations to have an effect. When we use the non-encapsulated encapsulated plant growth regulator, you can have a controlled release. So we can have a long-term effect with less application and a better response and a better production. So we are working with different kind of plant growth regulators, like nitroxide released chitosan nanoparticles, gibberellic acid loaded chitosan and alginate nanoparticles, auxine loaded zein and lignin nanoparticles, and also abscisic acid loaded zein and lignin nanoparticles. I will focus mainly in the case of the nitroxide here, just to give you an example of application. Next, please. So nitroxide, NO, is a signalling molecule in plants that has many functions in plants, from seed germination to stress resistance, root development, flowering control, and many other functions in plant growth and development and stress responses. But the application of this compound in agriculture are still scarce because nitroxide is a gas, so it's difficult to treat plants with a gas. So we use nitroxide donors, for example, S-nitrosophiles that we can see there. So S-nitrosophiles, they can be used as a donor of nitroxide, of NO, but they are very sensitive to light, to temperature, and also to the presence of metals. They release the nitroxide very quickly in these conditions. So we encapsulated, next please, we try to encapsulate the nitroxide donors, use chitosan. So chitosan, that is also chitosan is a very good polymer because it's a biopolymer that is produced from chitin, that usually people produce the chitosan from crustacean residues. So it's a natural polymer that's not toxic and so on, and biodegradable. And this chitosan could protect the nitroxide donor. It could allow a controlled release of nitroxide, as you can see there. So the non-encapsulated nitroxide donor release the NO more slowly, and it effectively increases the nitroxide content of the plants. Next, please. And we observe that the use of these nitroxide release chitinous nanoparticles improve the resistance of different plants to different kind of stresses. For example, we have there maize plants under salt stress. We had a increase in resistance. Also, soybean plants growing under copper stress, soil contaminated with copper, and also neotropical tree seedlings under drought stress. So the use of this technology improve the stress resistance by improving root growth and morphology, antioxidant response, and also the leaf gas exchange. Next, please. Next, you can go the next to go quickly. We are also not only worried about understanding the applications. Not only we are applying this to crop and seedling production for reforestation, but we are also interested in understanding the mechanism of interactions of these nanomaterials with plants by using fluorescently labeled nanoparticles, so you can track the nanoparticle in the tissues, in the roots and the leaves, and also you are using radiometric techniques also to track the active ingredient in the plants. Next, please. We are also using the seed nanopriming as another technology to improve the stress resistance. In this case, we do the treatment of the seeds with the nanoformulation. So the idea is when treat this priming. So we have this priming effect of the treatment of the seeds. So we observed, for example, with different species that treating the seeds with the nanoparticles, we could have a long-term effect in the stress resistance. Next, please. You can go also to the other. The next also. Just another. Just to go quickly because we have to. Not so many time. So it was about the nanoparticles. I would just like to tell you something about these composite biomaterials. That's another nature-based solution that we are testing. So in this case, we use agricultural wastes to make different biomaterials that could be applied to seed production and to agricultural production. Like we could do like some tubes, some pots, biodegradable pots to replace plastic. Also some biodegradable foams for nutrition purpose, hydrogels, and so on. Next, please. Just to give you some examples, these are the foams, the biodegradable composite foams that are made from, next, please, from different agricultural compounds like starch, lignocellulose fibers, glycerol, and so on. And they show to be very effective in improve the growth of tomato plants in field capacity and also in drought conditions. So it was very effective in increasing the plant growth in different conditions. Next, please. And also there are some micromaterials that has been developed. In this case, the materials are made from old husks, foam, and also yeast extract, and also this fungi, Panus strigelus, that's a native fungi from native fungus from Brazil. So these materials can be used also to the next piece to improve plant growth. As you can see, the next. The experiment with maize that we could increase the growth of maize plants, use this micromaterial, and something very interesting regarding these composite biomaterials. They can be used to provide nutrients to the plants, but they are completely biodegradable. But you can use these materials as a carrying systems for the nanoparticles, for the associative microorganisms, or for plant growth regulators. So they not only can provide nutrients for the plants, but they also can carry other microorganisms and other materials. Okay, I think the next. And we are validating, not only you are carrying out experiments in vitro and also in greenhouse, but you are also validating these different nature-based solutions under realistic conditions in the field also. And the next, please. And we are, as I mentioned, we have these experiments not only here in Paraná State, also in São Paulo State. We have collaborators in other places in Brazil, and we are also involved in some international networks, like in Europe. In Europe, with Biodiversa Plus, we have collaboration with researchers from different countries, from European countries, mainly France and Germany. Also, with last week, we had a mission of researchers from Australia and Mozambique to Paraná State, and they are also involved in this network, and also in some Latin America countries, mainly Chile, Argentina, and Mexico. So we are really open to collaborate also with the Japanese, with you from Japan, to join this network, to test these different nature-based solutions, to improve the growth and also the stress resistance of plants for agriculture and also for forest restoration. Okay, so I think it just the acknowledgements of the different, the next please, of the different funding organizations who, especially Quer, which is the main funder of our research, and also other national organization and international organizations. And thank you also for your attention. The last slide, please. Just you have my email here, and also some information about our projects. Thank you.
Unfortunately, our time is not so long. We have a short time.
Yeah.
Thank you for your collaboration. And they said me here that we have to finish until 10:45 to give some time to the Japanese delegation to now. So, well, it's tough, but I appreciate your collaboration. Thank you. Yes.
Good morning, everyone. Thank you for the invitation to be here to present our work that we do at Labim. My name is Rafaela Cardoso. I am a postdoctoral researcher at Labim, and today I'm going to talk about our laboratory, R&D, partnerships, and innovations. So our research teams comprises Admilton as a coordinator. We have here some postdoctoral, PhD, master students, and also some undergraduate students. And we also have some partners that helps us to achieve our goals. So our research group focuses on developing sustainable solutions for agriculture, food production, and other industrial needs. And here we focus on applied and basic science to support sustainability in different areas, such as biological control of pests, microbial diseases, promotion of plant growth, and production of bioactive microbial metabolites. So here we have an overview of all the areas that our laboratory have. First of all, we have bioprospecting. Here we isolate and identify microorganisms that could have potential to be applied in agriculture. Then we have microbiology and bioprocess. Here we do laboratory-based experiments and some scale-up bioprocess. Then we have molecular biology, genomics, and bioinformatics. So we use these methods to study the genetics of microorganisms and also identify potential genes or metabolites produced by them. And we also do downstream process as purification and identification of metabolites. Here we identify compounds and molecules produced by the microorganisms, and also study the formulation of the final product. And the final step, we have the validation step, that we test our microorganisms in real conditions, such as greenhouse or field conditions, aiming to prove that our microorganisms have the potential to be applied in the agriculture. So here we show the technology readiness level that help us to understand in what level of maturity our projects are. So here we have three main stages. Stage 1 comprises university stage, that here we do basic research. Here we have some activities. So as prospecting of microbiome inputs, we use genomics and bioinformatics. We do phase 1 bioprocess in the benchtop bioreactors. And we do also a chemical studies. Then the stage 2 comprises collaboration step that we do in collaboration with companies, and includes technology development, downstream prototyping, and formulation of the final product. Then we have the final stage, that is the industrial phase, that we transfer the technology to the industry. So here we have some activities as R&D in industrial bioreactor, regulatory licensing, and the registration of the final product. So here in this slide, we have an overview of all the activities that our laboratory do. First of all, as I said before, we have bioprospecting. Here we also do preliminary biological tests to prove that our microorganisms have the potential to be applied in agriculture. Then we also do performance validation in real conditions, such as greenhouse or field conditions. We also development of bioprocess for industrial applications. We do the identification of microorganisms using genomics and bioinformatics. And the phase 3 comprises the industrial phase, when we scale up for industrial applications into the final product. So here it's important to highlight that not all our projects goes to phase 3. Some of them stops in phase 1 and comprises only a basic research, and a few goes to phase 3 and become a real product, final product. So here we have an example of a project that we are developing in Labem called PGPR mining project. That is a project that involves a machine learning, integrating all the data that we have in the laboratory, including applied microbiology, bioprocess, genomics and bioinformatics, purify metabolites, and performance in real conditions. So this tool aiming to comprise all this data, aiming to identify the microorganisms faster and more efficient. So now I'm going to show some technologies developed or under development at Labem. So first, I wanna highlight Frontier Control, that is a biofungicide produced in development with the Symbiosis company, and this product uses as an active ingredient a microorganism that we isolate in Labin, and it's against Corynespos accacicola and other fungi. And this product has been on the market for 2 years, and it is estimated that this year it will reach 1.5 million hectares of planted land. So here we have another example that is a biofungicide development with Leaf company, manufacturing by solid state fermentation and in a wearable power formulation against a wide range of microorganisms using Talaromyces stolli fungus. And now these products are in licensing phase. Another case that we have in our laboratory is the development of Demeter, that is a product developed with FMC company that is against a wide range of plant pathogens, and is also using a microorganism isolate in our laboratory, Bacillus velezensis, as an active ingredient. And this product is already in the patent license phase. We also have a partnership with Vitalis that we are developing a multi-site biofungicide that is using, once again, Bacillus velezensis as an active ingredient against some fungi. And now these products are in the licensing phase. So to conclude, I want to talk about a business case that was born at Labin. So Bio3 is an academic spin-off with a business model of a startup created by members of Labin that have the focus and the objectives to do research, development, and innovation for agriculture. So here we have a case that we integrate the science, the basic science, to the industrial needs. So thank you for your attention. I appreciate the invite to share a little bit that we do at Labin. This is our contacts. Feel free to reach out, and thank you once again.
Good morning. Ohayo gozaimasu. My name is Gabriela. I'll be presenting on behalf of Professor Galdino. We're from the Microbial Ecology Lab in here at WELL. And I'll be talking about phytopathogenic fungi control in arbuscular mycorrhizal fungi. I'll try to be brief. Pode começar. 10 minutes, okay. So, to use a microorganism as an antibiotic, the basic steps are cultivating the strain of interest in nutrient broth together with copper and water for 10 days at 28 degrees. And then moving on to be centrifuged for 20 minutes at 4 degrees Celsius. And And then we need to do an initial test with the supernatant from that centrifugation. So we can test the antibiotic effect with a Kirby-Bauer test or disk diffusion. Briefly, we separate then into a liquid-liquid partition, the antibiotic fraction with organic solvents, and we move from the least polar to the most polar substance, and then we move on to different separations. So vacuum liquid chromatography is the next step, where we obtain the F3 fraction, which will be later on even more purified, so we can obtain the F3D, which is our fraction of interest that will test the antibiotic effect. So this is a basic flow chart of how we obtain the F3D fraction. And this F3D fraction is tested again for its antibacterial effect, this time with a thin layer chromatography and bioautography. And then this F3D fraction was, after a few experiments, needed to be a little bit more refined. So we went through more processes of purification. So we obtained this even more purified fraction called F4A. And it contains basically phenazine carboxamide, phenazine hydroxylamide, indolin, and metal antibiotics, which will be our substances of interest. And then we can test this antibiotic in against different fungi. For example, the Phacospora pachyhisi, which is a soil-destructive fungi. So the test in vitro, it consists in concentrations of 1, 10, and 100 micrograms per milliliter. And we will observe its effect in vitro. So we will evaluate the spore germination and germ tubes growth in vitro. So this is the control. We can see how the hyphae are, how it's germinating, there are many hyphae. And then we can see as the concentration increases, how we have less germination and the absence of hyphae. And then we can test that on the field, and how we do that is through cultural treatment. So we use a combination of sphere, Unizeb, and then combinations of only the fraction of interest, the F4A, and combinations of F4A with sphere and F4A with Unizeb. And we do 4 applications each of 200 liters per hectare with concentrations of 1 and 10 micrograms. And how we evaluate that on the field is through crop production. And we can also monitor the disease progress through a under the area disease progress curve. There were also some tests were also made with collected leaves from the area, and we ran into this study that evaluated this. They scanned the leaves in an electron microscopy, and here are some images. So this is the control group. We can see how all the structures are intact from the fungi. So we have hyphae, we have spores. And then as we move, pode ir passando. And as we move, pode passar. And we can see how the structures begin to be, so we can see how the spores on the second image, it is a bit destroyed, and how the hyphae become compromised as well. Pode passar. This is also a bioautography test that was done with this F4 fraction. Pode passar. Some disk tests as well, disk diffusion. Pode passar. Pode passando. And this is the minimum inhibitory concentration test that we did. Pode ir passando. And you can see as the concentration increases how it is effective against the fungus. Pode passar. And this is in the field test. We can see we cannot see, but the biggest production. So the highest bar is actually the leomycin 20 micrograms versus the first one that is the standard product for this, the Fox by Bayer. So we can see how more using the leomycin 20 micrograms, how it's more effective than the standard product that is used usually against this fungi. So it increases production of cotton. This is the phenylaxine. So we can see the last bar is with the highest amount of production versus the Veritas, which is also a standard product that is used on the field commonly. Pode passar. There are some tests with using Bolcodaria against the Cicatoga fungus. Pode passar. Pode passando. Now there's some images of this Bolcodaria against different types of fungus. Pode passar. Pode passando. And here you can see this is antagonism between Sclerotinia and Pialomyces. Pode passando. And here we go through insecticidal activity of some symbiotic bacteria found in the Heteroharbidites against Galeria. And in the images, we can see how using this bacteria that is found in this larva, how it causes loss of body turgor and also melanization, compromising the life of these insects. Pode passar. And here, with the largest concentration, the P4, we can see how the survival rates drops by a lot with a high concentration. Pode passar. And here, the P4 extract, which is the middle image, we can see how it compromises the size and the health, I would say, of the animal. And this P4 extract is the one with the highest concentration. And this test was done by spontaneous feeding. So combining artificial diet with ethylacetate extract from this larva. Next. But pode passar. These are some fungi that were isolated and tested against some insects as well. Pode passar. You can see how it's effective in infecting these animals. Pode passar. And here again, so each one of these F11, FI2, they're all different fungi. You can see how they're effective in infecting these insects. So it's a naturally and commonly found fungus in nature that can be used against some very agronomically important insects that destroys a lot of crops. Pode passar. Here's some survival. You can see how the survival drops with the use of the different fungi as well versus the control group. Yep. Pode passar. And finally, about the mycorrhizal fungi, we work a lot with that in the lab. And the stages of development, as well as was mentioned by some other professors here. We do 3 stages. So we multiply them in lab, in the lab with roots, using roots. Modify carrot roots, so we inoculate them, and this is how we multiply them in lab. And then the second stage, we test that in the greenhouse experiments with different agriculture, different species of interest for agriculture. So soy, cotton, tomato. And as the third stage, we use that in the field. So we do run tests in the field. So these are images from the lab. This is the evaluation of the colonization potential of Rhizophagus clarus, inoculum produced in vitro. So it's a species of arbuscular mycorrhizal fungi. So the first phase of every image is the control group. The second phase in every image is the plant that was inoculated with the mycorrhiza in the vase. So in the moment that we planted the plant there. And the third phase of each image is the soil was already inoculated with it. So you can see how effective it is in its growth and health. This is the plates where the petri dish where we, using this transformed carrot roots, where we inoculate the mycorrhiza and how we multiply them in lab. These are some studies that were conducted by students in the lab as well. And you can see how, so they tested the effect of the arbuscular mycorrhizal inoculation in grain yield of 2 different cultivars of soybean. So 133 and 359. And so we have the control group with no fertilizer and no mycorrhiza at all. And you can see how the combination, the group that has the most, the largest production, which is the fourth set of bars. You can see how the combination of mycorrhizal and fertilizer was the most effective in production. So this was tested for 2 different cultivars of soybean. And this is, the combination, as I said, the combination of, it's a Rhizophagus clarus with combination with fertilizer. Next, please. And this was also tested in cotton yield. And you can see how the control group with no fertilizer or urea versus the T4, which is the most productive batch with the most productive group. And it was a combination of Rhizophagus clarus with PK and urea. And having said that, I think that discussing with the professor, we thought that good collab ideas between Brazil and Japan could be to maybe chemically produce these molecules of interest, or help us or do the study behind the pharmacodynamics of these molecules of interest. And thank you very much. Arigatou gozaimasu.
Okay, thank you. So I think we have short time just to last presentation, right? Just to have time to discuss with the Japanese people. I think this is also very important, just to see what we can do in the future.
Good morning, everyone. It's a big pleasure to be here with this Japanese research group. I'm waiting. I will be very brief because it's at the time it's very rush. So I will try to explain a little bit about functional foods that we conduct here in State University of Londrina. My name is Professor Carla Bijete Gergoleto. I am from the Department of Food Science and Technology. I am Vice Coordinator of Postgraduation in food science. So I will try to explain a little bit about my results and from my colleagues as well. I started already because they are having a little bit problems here, but yes. Mhm.
Ah, não, mas o dela é no...
Deixa eu ver. Maybe. Just because we are in rush, the presentation are not helping us.
Chegou aí.
Oh, thank you. Oh, ufa. Okay. So thank you very much. I will try to be very, very brief on my presentation, just to you to know what we are doing here in our department. I will start my presentation showing my colleagues from our department, mainly Dr. Elisa Hiroko Yoka. She already has a lot of contribution with the Japanese projects, and she works with functional foods and also with food security. We also have Dr. Adriana that works with meat production, Dr. Vilma with stingless honey bees, Dr. Sandra Prudencio that works with bioactive peptides and sensory analysis, and Dr. Thais and me. Mainly our research area is we focus on probiotic fermentation products. We try always to include native fruits with high antioxidant potential. These native fruits, it's became very interesting to work beyond the consumption of it all, but trying to substitute some ingredients to these native fruits. We also study bioactive peptides and single beet honey. I will introduce my research very, very, very brief. The main research of my area, it's with Jussara pulp. Jussara belongs to the Atlantic Forest region. Its pulp is very similar to acai pulp. But the difference is because we have here in our region. And Jussara pulp has a lot of antioxidants, more than acai fruit. This palm tree is becoming extinction because they illegal in extraction. So this research is conducted with different areas, with biology, scientists, agronomy, agriculture, because we want to introduce the Jussara pulp to our consumption, regular consumption. But for this, we have to produce this fruit in a kind of sustainability. We already have a lot of works done with the potential of Jussara pulp with probiotic bacteria or not, being in fermentation, gut fermentation. The Jussara pulp can modulate our gut microbiota. It's bioaccessible even after the in vitro digestion. We can do a lot of products with Jussara pulp. I brought here the spray drying process that this pulp is very perishable. So the dehydration by spray dryer, it's an alternative to produce this pulp. And besides the dehydration, we lose a little bit of these beneficial compounds. They still remain with a higher antioxidant activity, even if they are in dehydrated form. We also conduct in vivo study with this Jussara pulp. We evaluate the Wister rats in advanced reproductive age, and we observe that these rats that receive Jussara pulp, they had an increase in fertility and a decrease in pre-implantation loss. We also observed a less damage in ovaries and uterus tissue. So demonstrating in vivo the potential for Jussara pulp to introduce for advanced reproductive females. We are already conducting the development of new products, mainly with kombucha, saborized with native fruits. The main of this product is because we have here a lot of red fruits, but coming from other countries. And we have here in Brazil very important fruits like a Jussara, jabuticaba, grumichama. They are produced here, they are from here. So we are developing products from our native region. This product is already established. The consumers, they have a very good acceptance by consumer, and now we are starting to conduct the in vivo studies. This kombucha already produced is being administered by football players. They are drinking 200 milliliters for each day for 90 days. And we are evaluating their performance, immunological, oxidative, metabolomic, gut microbiota. And this is a research being conducted with the sports science as well. So for us, it's important to develop it, to study these functional foods, but to complete with these in vivo studies to see how is it. So we are also working with these native fruits to substitute chemical additives in meat products. So Dr. Adriana is substituting, reducing salt, reducing additives or antioxidants, synthetics by these fruits, these residues. And we saw that these products presents high stability during refrigerated storage. They have antioxidant potential similar to synthetic ones. They are darker than the products, normal ones, but they are very well accepted by the possible consumers. We also have studying with the stingless bee honey together with Dr. Caltarelli. Here we are working with the development of the products and the introduction of these products in human trials. So Dr. Vilma and Dr. Sandra, they produced a candy using organic and traditional production of honeybees with or without probiotic bacteria. And they gave to teenagers to observe the oral microbiota. And the teenagers that received this kind of candy made with this honey reduced the dangerous microbiota present in oral mucosa. And being with peptides bioactives, we are studying different kind of productions of peptides bioactives. For example, I brought some beans, germinate beans that increase the production of peptide bioactives. And Dr. Thais is developing a flower of these germinate beans with higher peptides bioactives, and producing cookies, bread, and evaluating if their peptide bioactives can reduce ACE inhibitory enzyme, for example. And we are also evaluating these peptides bioactive with some bioinformatics to reduce costs before going to human trials. So it's a very brief that I prepared before. I'm sorry for that, but thank you very much for your attention. A special thanks for the results from Japan, and I hope we can show a little bit about our results, and we are very pleased to receive you in our group to do novel collaborations, to discuss new ideas. Thank you.
Dr. Jackson, we use the little time we have to make some discussion.
Yes, I think this is one of the most important time. I invite Dr. Watanabe to conduct some discussion, presentation, and some questions as well. And thank you, Professor, to the last presentation, just to shorten, and because we are in short of time. So please, Professor Watanabe.
Okay. So the time limit, one thing, the Professor Tamura, I think will come back, right? After, 今日だけでなくて、いっぺんクリチバイというかこちらお戻りになられますよね。Professor will come back after visiting Curitiba, then he'll come to again Londrina. So you'll have the specific discussion over the functional food. So number one. And number two, so thank you very much for the sharing the quite progress on your the research activities, generally speaking. And let me take some points. So bioremediation, biocontrol area, likewise you do, we have the several research groups, depends on discipline and the target. So it is likely your interest to match with them. This time I don't take any microbiologists or mycologists here, but actually we have many. And we have like a nationally appointed research center. So that was one area. And there was a suggestion on your research result application of pharmacological aspect. And again, we have those of series of research on the human cell evaluation system and the mouse-based, like a knockout mouse, the genomated mouse-based evaluation, and the one more step at the potentially clinical applications are the systems. And actually, the group of the medical and the pharmacology group, they are quite interested in Brazil. So they have some activities with USP in Sao Paulo, and the president of the university also on the pharmacy. And particularly his group also has interest with the state of Paraná. So I come here to make sure if we can match with your interest, and yes, surely we can have the further step to introduce our group, okay, to your group. So, and Dr. Kanamori has time, a few minutes to make some of the comments.
Yes, I'm Norihito Kanamori from JIRCAS, Japan International Research Center for Agricultural Sciences. So I was work at the Envelope Soybean Center for 6.5 years and focus on the what is it, for the plant soybean breedings and soybean GMO soybean for tolerancia seca, drought stress tolerance. And so we also have another researchers for the soybean breedings for the soybean rust. And also we also have the many researchers on the soybean and blood rhizobim and the soybean mycorrhiza interaction symbiosis. So we have many researchers, and so today's in slide we can see many some harmonize of our research, and we hope that is a quite good collaboration for the future, I think. And I also think we don't have some research about, for example, the such as nano primary coating or some another technique. So that is for we would like to know more detail. So that is quite a good time to start our collaboration, thinking our collaboration. Thank you very much.
Okay. So a couple of additions. So the particle nanotechnology aspect, we have within Tsukuba is a science city, and we have 174 institutes. National Institute of Private Company Research the organizations. So one of them was material science. And we have the medical area collaboration and also agriculture area collaboration over how to use nanotechnology. So we can bridge your group with the acronym is NIMS, maybe the known also here. And so we can make like a tri-party, four-party collaboration possibility exploitation. So I'll be the contact point for you people, and I'll make sure that you have the correct communication with the different institutions. And finally, thank you very much for introducing different native fruits, the utilization. We know already and commercialize the Acerola Cam products in Japan. We're happy with that. And now we are looking at how much information we will get from other fruits like jabochikaba. And actually, Japanese interest wants to import, but they want to have more scientific validation information, how it works, how you enjoy, and they would like to come. So the research-based the commercialization, actually. So that's what our side of private sector would like to see that. Okay, thank you very much. Hi. Hi. すいません、ドクター田村ですね。
My name is Hirotoshi Tamura, come from Kagawa University, and my specialty is very close to the functional food. Then But this is a very interesting meeting because not only the chemist, also the economist is joined together today. And then point of view of economy from my side is maybe the establishing some rule of what is a functional food. If I see the Japanese side or government decision, before the 1980, we just report what is important in foods or food material or natural products. Then just to see, oh, this is important. But after that, Japanese government decide the level of importance or likely medicine level. We must investigate a lot to get the evidence of importance. It's the highest, nearly like a medicine itself, but come from food. We are really appreciate of safety and reliability of data. That's why government contribute to the making a rule. The second level is just of the nutrition value, some mineral and vitamin C is very important for our body, of course. This is already established. Just accept the important level of concentration in food. In case of acerola, it's a rich of vitamin C, but we are requirement is 100 milligram level a day. If over that one, already importance of our health. That's why we can display the importance in the packaging. This is a very reliable information for consumer. Then third one is just company itself can get the evidence from scientific review. Just they don't research so well, but can get the information from scientific papers. Then collect the importance data from papers, and then propose it consumer affairs of a kind of government. Then every small company can display the importance of any biological activity. Then it is not curing problem of disease. We are just want to keep health condition. This is enough. This is an important level, I think. Everyone needs health condition. Just a comment. Right now, by display on the packaging, the level of consumer demand is quite increasing a lot. Maybe likely 60 billion United States dollars are required or consumer or buying such important health claim materials in the market. That's why if we were establish such another approach to making a rule is a very, very good condition for your further collaboration, I think. That's all. Thank you very much.